This is the cell cycle. The cell cycle is made up of Anaphase, mitosis, and cytokinesis. Now let's look at why cells divide. The reason that most cells are so small is because the size of the cell determines how much food it can take in and at what rate. When the cell is small, diffusion can occur much faster because there isn't as much surface area and volume that materials have to pass through. In larger cells, diffusion becomes slow and inefficient. You can compare this to the college admission process. Small colleges tend to accept people at a higher rate and faster too. But for large universities like UNC, they have a lot more applicants and their acceptance rate is lower. It also takes a lot more time and resources to complete the process. Large cells are like UNC in the fact that it's harder for them to perform diffusion at a quick enough rate. Another reason that cells divide is that the bigger the cell, the more proteins that need to be produced. DNA only codes for so much protein to be made. When the factory cannot keep up with the demand for protein, the cell must divide. The first stage in the interphase is G1. This is the longest stage, where the cell is growing. Here we find the G1 checkpoint, where the cell determines if it is big enough and if the environment is right in order to divide. If the answer is yes, we move on to the next stage. Now we move into S phase, which is the second stage in interphase. Here the cell replicates DNA. First, the enzyme helicase unwinds the double helix. Next, DNA polymerase attaches free nucleotides to the template DNA strands. The result is two double helixes of DNA. These two identical sets are known as sister chromatids. Next, we go to the last stage within interphase, which is G2. Here, there is another checkpoint. The cell checks for any DNA mistakes, if the cell volume is adequate, and if the environment is suitable. If it is, we move on. Mitosis is a process of cell division. Cells have to divide in order for an organism to grow, repair damage, and replace old worn-out cells. Mitosis has 4 stages: prophase, metaphase, anaphase, and telophase. In prophase, chromatin condenses to become chromosomes, then the nucleus begins to disappear, centrioles move to opposite sides of the cell, and lastly spindle fibers form. The second stage is metaphase, where chromosomes line up in the middle of the cell, then the nuclear membrane is gone, centrioles are at opposite ends, and lastly spindle fibers attached to the chromosomes. The third stage is anaphase, where chromosomes are pulled away or apart from each other by spindle fibers, and there is still no nucleus. The last stage in mitosis is telophase. This is where chromosomes are separated, the spindle fibers disappear, and the nuclear membrane is back. The last stage in the whole cell cycle is cytokinesis. This is the division of the cytoplasm inside the cell to make 2 daughter cells. When things go wrong in the cell cycle, it is because the cancer cells ignore the signals that regulate the cell cycle. Tumors come about when cancer cells keep dividing and form a mass. The 2 types of tumors are benign, which are abnormal mass of cells at the original site, which is normally not harmful and can most of the time be treated. Then there is malignant, which is what people worry about because it is a mass of cancer cells that impairs organ function. The causes for cancer are cigarette smoke, UV light, radiation, chemicals, viruses like HPV, and genetics. For the treating of cancer, the 2 main ones is radiation, which is high energy radiation that disrupts the cell cycle to stop the cells from dividing anymore. And then the last one is chemotherapy, which is the drugs that disrupt mitosis.